If you're watching this video, you're probably aware of a particularly niggling and insidious rumor that has been spread all across California and all across the United States. And the permutations that I've heard go like this. Broad Mite and Hemp Russet Mite are, or have been, spread by the California Department of Transportation as a biocontrol agent for pest weed species, and that it is also a surreptitious plan on the account of the government to undermine cannabis growers by spreading these pest species. And I've seen on social media, particularly Instagram, which is the catalyst for this video, that this report is it's a cited source that people are saying explains and articulates that these organisms are being spread for nefarious reasons, and I don't agree. I've read this report. I have not read every single word, but I'm going to do my due diligence. I'm going to try to bring my own commentary on this report. I will probably post a full kind of raw extended version of this report, of my commentary, and an abbreviated version. And mostly because I have a feeling that this 24-page document is gonna run me a while. So anyways, that's sort of my introduction here. This is a video that is commentary on the technical report titled Enhanced Biological Control of Yellow Star Thistle and Tumbleweed, Russian Thistle in parentheses. And so let me go over this, right? Here we have the first page. I'm going to briefly go over some important details. The title is Enhanced Biological Control of Yellow Star Thistle and Tumbleweed. Parenthetical statement, Russian Thistle, right? The author is a Dr. Lincoln Smith, and the performing organization is the U.S. Department of Agriculture, Agricultural Research Service, the Exotic Invasive Weeds Research Unit, Western Regional Research Center, 800 Buchanan Street, Albany, California, 94710. And the report date is July 9th, 2006. I am recording this audio and video in 2017, September 13th, in fact. So it's more than a decade since that time. The sponsoring agency here is shown to be the California Department of Transportation, Division of Research and Innovation, MS-83, 12270 O Street, Sacramento, California, 95814. And the type of report and period covered is final report, and they don't show the period covered for some reason. But this is a final report on a assessment and evaluation for mainly 2 biological controls. And we'll get into that later, but I just want to point out here boxes 15 and 16, supplementary notes in cooperation with the U.S. Department of Transportation and the Federal Highway Administration, and the abstract, which is one of the most important parts of this whole document. If you don't read the whole document, if you don't watch this whole video, at the very least, listen to this abstract. Yellow star thistle and tumbleweed, Russian thistle, are 2 of the most important weeds on California Department of Transportation, CALTRANS, rights of way. They occur in high densities, outcompeting desirable grasses, and increasing risk of wildfire and soil erosion. Transportation rights of way serve as corridors that can aid the spread of weed seeds by vehicles. Weed-infested rights-of-way can also be a source of seeds that might contribute to the spread of weeds into neighboring properties. Both these plants are alien to North America and have previously been targeted for biological control, but the introduced agents have not reduced these weeds in California. Release of new agents can provide widespread and perpetual reduction of weed populations without further expenditures. So right away we see that similar and matching with the rumor, CALTRANS is trying to find biological control agents to control the spread of 2 weed species, and neither of them are cannabis. So, but the 2 species that they talk about the most here are an Eriophyid mite, to which russet mites of all stripes belong to. At least it's one group in Eriophyiodia, which is not the same as Eriophydia, but anyways. And the other one is a weevil species, so that's a beetle, right? I wanted to point out that it seems like they're trying to use multiple kinds of biological control to control these plants, and they've learned from previous mistakes in the world to use biological control agents. So it's not like they're being irresponsible here. It seems that they've got an expert and a whole unit of experts working on this problem, and they check their work very detailed, very careful. Besides only taking a cursory glance at a couple of pictures and a couple of key words, I don't see how somebody could read this document and make any other conclusion than CALTRANS did not put out russet mites or broad mites. In fact, broad mite, the genus for broad mite, so broad mite's scientific or binomial name is Polyphagotarsonemus. Latus is the species name, and that basically means many-eating tarsonomid. And tarsonomid is the family of mites that broad mite is a part of. Thread-footed mites, they're called. And that would be an incredibly insipid thing to do. It's a pest on many agricultural crops, many horticultural crops. Deploying broad mite as a biological control for these 2 weeds would be terrible for the economy and terrible for global agriculture. So I have a hard time believing that they would be doing that. So there you go. And then this document, you will not find a reference to Aculops cannibicola. You will not find Hemp Russet Mite referenced anywhere in this document. You just won't. But you will see a picture of an Eriophyid mite, and that mite does have a morphology consistent with russet mites because they're vermiform, and they have that characteristic sort of tapered worm-like body. And I imagine what has happened is that people have skimmed through this document, which I don't blame them for doing, and then they see this picture, and people who perhaps, they're struggling to find information, they're stressed, they're maybe not, they may not be phytopathologists, right? They might not be insect experts, so, or in this case, they're mites, right? So they just might not know all about pest physiology and taxonomy and all that stuff. And I'm not trying to disparage people who don't, because that's understandable. I don't know about a ton of different disciplines, but this is in my wheelhouse, and I thought I would talk about it and sort of assuage some of the concerns that I hear on the topic. So, without further ado, I will continue on this commentary, right? So, here, We've got a few keywords that I'll go over. Host plant specificity, classical biological control, non-target plant, risk assessment, yellow star thistle, tumbleweed, Russian thistle, wildfire, and weed control. I'm just going to touch on host plant specificity for a moment. That's an incredibly important part of biological control, especially for plants. It's one of the reasons why it's ludicrous to me that anyone would think that, seriously think that broad mites are being used as a biocontrol for these weeds. They would affect not just these weeds, they would affect other crops. I mean, I don't know how there's multiple ways I can say this, but really, plainly speaking, it would be a bad idea. Capital B, bad. And I know that host plant specificity is an important evaluation, is an important aspect of evaluation for biological control. And here we have a distribution statement. No restrictions. This document is available to the public through the National Technical Information Service in Springfield. So anyone, including myself, is able to download this page and read through it. Not everyone has read through it appropriately, and that's why I'm doing this video again. Not to be a broken record, but I'd like to really reiterate that there really is no evidence for Hemp Russet Mite or Broad Mite being used. There's not. If you find it, let me know, because I might have missed it. Alright, so we have here enhanced biological control of yellow star thistle and tumbleweed, Russian thistle. Final report by Dr. Lincoln Smith, U.S. Department of Agriculture, Agricultural Research Service, Exotic Invasive Weeds Research Unit, Western Regional Research Center, 800 Buchanan Street, Albany, California, 94710, 9th of July, 2006. It bears mentioning, right, that the U.S. Department of Agriculture, the people who would be aware of these pests, broad mite in particular, were a part of this research. There's a disclaimer statement I'm not going to go over in great detail. Here's a table of contents. I'm also not going to go over in great detail. List of tables, list of figures. There's an executive summary. I'm going to go over this. There are parts of the report in which they reiterate some of these points over and over again. I'm assuming that they do this for the same reason a lot of government documents do this, is for redundancy, so that people don't miss important information, and because it's relevant, it's relevant every time you bring it up in different aspects. So let me go over it. We conducted evaluations of host plant specificity and potential efficacy of prospective insect and mite biological control agents of 2 invasive alien weeds, yellow star thistle and tumbleweed, which remember is Russian thistle. The biological control agents are alien to the U.S. and must be evaluated before it is possible to obtain permission to release them. So ostensibly, and I don't know if this is the case, because I haven't followed up, ostensibly, even if these were Hemp Russet Mite and Broad Mite, which there's no way they could be, there would have to be an evaluation, and there would have to be permission granted by other authorities before they could even release anything. If it was some kind of a black operation, and there was like a gag order or something like that, we probably wouldn't even be looking at this document. Or it could be plausible deniability, who knows? But once you start making that once you start using that as an excuse, I don't know, I would like to see some evidence of some kind, maybe some investigative journalism if people are still confident that this is the case, that the rumor is true. Yellow star thistle. We completed evaluation of host plant specificity of the rosette weevil from Turkey, and I'm only going to say this once, Ceratapion barsicorna, We tested 51 species of non-target plants, including 4 economic species and 24 species native to California. The results indicate that the weevil poses no risk to native plants, but it may cause some damage to bachelor's button, which is both an invasive weed and an ornamental flower. Bachelor's button is a flower that I've grown. They're also sometimes called cornflowers. You can get them at a ton of different stores all the time. They come in different colors. So this damage is no worse than that caused by other previously approved biological control agents. Under greenhouse conditions, weevil infestation reduced growth of well-watered, fertilized plants by 23%. Impact under field conditions could be greater. Obviously, because they're not always well-watered and not always fertilized, which means that they will be more stressed and the plant might be even less resistant to damage. We submitted a petition to the Technical Advisory Group, TAG. I'll often refer to this as TAG, but I might switch between the two in case I think it's important for context, of U.S. Department of Agriculture, Animal and Plant Health Inspection Service, which is the USDA APHIS, is how I'm going to refer to that, for Sarah Tapion Barsacorna in January of 2006. TAG has representatives from all the U.S. federal and land management agencies. Canada and Mexico are included. The petition is still being evaluated by TAG. No release can be made until APHIS issues a permit. In the meanwhile, we began evaluation of the flea beetle, which is Psiloides calcomera or chalcomera, depending on how you want to pronounce it. I'll say it right now. Latin is a dead language. There is a correct way, certainly, but unless you've taken Latin, sometimes those correct ways can be quite esoteric. So sorry if I make any pronunciations incorrect. Let me know. I do like to say things right if there's an established way of saying it. And Psiloides calcomera is from Russia. We have tested 55 plant species and expect to complete testing this summer. We produced a short manual on biological control of yellow star thistle, which is in Appendix 3. Tumbleweed. We completed evaluation of host plant specificity of the Blister Mite. I'm going to stop right here and say that Blister Mite is a common name for a lot of russet mites. A lot of Eriophyid mites are called blister mites because of the, or they're sometimes called gall mites, and they can create these leaf malformations. Sometimes they're galls, sometimes they cause the leaves to curl, right? They have these effects. So this indeed is a blister mite, and there is a photo of said blister mite down in the report, and I think a lot of people looked at that picture and they kind of they saw what they knew was a russet mite, or what they thought to be was a russet mite, and I think that's where some of this rumor comes from. Anyways, Blister Mite from Greece, and it's Aceria Salsolae. We tested 36 species of non-target plants, including 6 economic species and 25 species native to California. The results indicate that the mite only multiplies on the target weed and its close weedy relatives. Under laboratory conditions, mite infestation reduced growth of well-watered, fertilized plants by 66%. 2/3 is not bad. We submitted a petition to USDA APHIS, Technical Advisory Group, for Aceria Salsolae in December of 2004. TAG recommended approval of the mite for release in August of 2005. We wrote a draft environmental assessment and submitted an official request for a release permit to USDA APHIS in September of 2005. The permit application is still being processed. No release can be made until APHIS issues a permit. In the meanwhile, we continued evaluation of 2 other candidates. Tests in France determined that the bug Piesma salsolae, which is funny, has the same species name, is not specific enough to be safe for release, and work on this agent has been discontinued. So that's important. I want to reiterate that. Some people have it in their minds that that certain government bodies, especially those related to agriculture, don't do their due diligence, and historically that happens. I won't deny that, and I'm not trying to be an apologist or anything like that. But here we have direct proof that, this Dr. Lincoln Smith shows that they were trying to find another insect in the meanwhile time, during their application process, and they found that the one that looked promising is not, because it will in fact infest other species. So they're not going to have a cane toad problem. We evaluated the seed feeding caterpillar, Gymnansila cannella. Gymnansila cannella. That's a nice name. For 3 years and find it is more specific. We hope to complete testing this fall. So they were able to find a lepidopteran that would be better than this flea beetle. That's how it goes, right? Steps required before making releases. This is important. I really want to iterate this to people who, like myself, before reading this article, are not necessarily aware of what the process is for this to happen. Number 1, scientist submits petition to the Technical Advisory Group, TAG. The Technical Advisory Group recommends approval to release. Scientist submits permit to the USDA APHIS. APHIS writes an environmental assessment and consults with U.S. Fish and Wildlife Service. If they conclude that there will be no significant negative impact, FONSI, then APHIS can issue a permit for the scientist to release the agent. The state of California must also permit this release. So there are multiple redundancies in this evaluation process, right? Before release can even take place, a ton of different bodies and organizations and people have to look at this and assess whether or not this makes sense. I have no doubt that people affiliated with the UC extension system would be part of this process, or people who are who have a similar sort of understanding. Again, this is a big part of why I feel like the Broad Mite thing just doesn't make sense. I know that sometimes the government can make poor choices, but man, this would be a very ignorant move. And it seems like they're covering their bases when they make their evaluations, like with the yellow Starthistle and the Tumbleweed paragraphs. I will also put this link down below in the description. The regulatory process can take 1 or more years, and the more information is at this link, and I will put it in the description for you. It's from the APHIS USDA government page for the Technical Advisory Group about permits. Okay, so I'm going to talk about the need for the project. I'm going to go over the paragraphs for the yellow star thistle and the tumbleweed, and also the intro paragraph here on page 2 of a lot. So bear with me, if you will. Yellow star thistle and tumbleweed, Russian thistle, are 2 of the most important weeds on California Department of Transportation CALTRANS rights-of-way. This is what I was talking about when I said that they often have multiple redundancy in their writing. So you're going to hear a lot of the same sorts of sentences and slightly different versions married with other sentences that are not reiterated. They occur in high densities, outcompeting desirable grasses, and increased risk of wildfire and soil erosion. Transportation rights-of-way serve as corridors that can aid the spread of weed seeds by vehicles. Weed-infested rights-of-way can also be a source of seeds that might contribute to the spread of weeds to neighboring properties. Both these plants are alien to North America and have previously been targeted for biological control, but the introduced agents have not reduced these weeds in California. Discovery of New prospective agents in Eurasia provides new opportunities to achieve successful long-term control of these weeds. The cost-benefit ratio of successful biological control projects is usually very favorable. Examples range from a 1 to 2 to a 1 to 1,675 ratio. Star thistle, yellow star thistle, infests about 14 million acres in California. Jeez. Primarily in rangeland and road rights of way. It is toxic to horses, causing the lethal disease nigropolital encephalomalacia. Reduces plant biodiversity, and the spines interfere with cattle grazing and human recreation. 6 species of insect biological control agents have been introduced to California, 2 of which are now widespread and are beginning to reduce seed production. The rust, Puccinia Yaceae, variety solstitialis, yes, I'm going to go with that, was first released in California in 2003 and is being released throughout the state by California Department of Food and Agriculture, the CDFA. Although the rust is beginning to infect the weed, it does not appear to be causing much damage. Developing new biological control agents that attack the root, stems, and leaves of the plant will complement the impact of the other biological control agents and will help to provide long-term control and reduce the need to apply herbicides. Right? So all this makes sense to me. This yellow star thistle is an alien species. It's an incredibly invasive species. It causes problems for horses. It causes problems for people trying to hang out. I'm imagining the spines are probably like thorns almost. I've definitely come across similar weeds more prolifically at least, and they're not fun to dig out of your skin when you're barefoot. Which I assume is what they're referring to. And they have biocontrol agents. They've got a rust here. They've got other biocontrol agents, but they just haven't been effective enough. So they're looking for something else so they don't have to spray herbicides. It's literally the reason why they're trying to do this, is so that they don't spread a bunch of herbicides. Tumbleweed, Russian thistle, Salsola tragus is the species name. And that's important because you might remember earlier that the Blister Mite species had a similar species name. And so did the flea beetle, I think. It is a major alien weed infesting roadsides and other disturbed sites in California. Wind-blown tumbleweeds careening across roads may increase the possibility of automobile accidents. I've heard and seen some people making commentary about how this is sort of, I guess the word would be ridiculous, that tumbleweeds could cause accidents, and that it's kind of maybe a dumb reason that somebody would be trying to control the weed, but there it is. I've never had problems with tumbleweed, but I imagine other people have. So there you are. That's one of the reasons. Tumbleweed is an important off-season host of the beet leafhopper, which transmits curly-top virus, which attacks several hundred varieties of ornamental and commercial crops, causing severe damage. Ain't that the truth? A lot of hemipterans are vectors for disease, pathogens, and viruses. So not only can this tumbleweed be a problem for, first of all, outcompeting other plants, but secondarily for automobile accidents. It's also a reservoir for a pest that has that is a host of a virus for many crops, and that's a great reason to get rid of this invasive plant, to be quite honest. Tumbleweed is native to southern Russia and western Siberia, but now occurs across much of Europe and Asia. 2 moths were introduced for biological control in the 1970s. However, predators and parasites prevent them from becoming abundant enough to control the weed. So, maybe against sort of conventional understanding of biocontrol, and invasive species, sometimes non-natural organisms don't establish. Sometimes you introduce them and they are readily met by the native population that will certainly predate on them. And it looks like that's what happened here with those moth species. Developing new biocontrol agents will help reduce both the size and number of tumbleweeds and reduce the need to apply herbicides. Again, they're trying to not have to apply herbicidal agents. They really want to use biological herbicidal agents, I suppose you could say. Increasing the effectiveness of biocontrol will help CALTRANS meet its commitment to manage these weeds and reduce usage of herbicides by 80% by 2012 without increasing maintenance activities. Wow. Again, I haven't followed up with any of this, but that's a lofty goal, and I congratulate them for trying to meet that commitment. I think that they sometimes don't get enough credit for trying to do things like that. So the project goal is to release natural enemies that are highly specific to yellow star thistle and tumbleweed, Russian thistle, to reduce the weed populations to innocuous levels. There are project objectives and achievements. I'm not going to go over the achievement as much, but I will go over the objectives here really quickly. It is to discover promising arthropod biological control agents in Eurasia, test them to determine if they are safe, test them to determine if they are likely to impact the target weed, obtain state and federal permission to release them, and provide information to CALTRANS on biological control of yellow star thistle and Russian thistle or tumbleweed. This paragraph here says, we evaluated the most promising agents, a root-feeding weevil for yellow star thistle and a blister mite for tumbleweed, in quarantine laboratory to determine their host plant specificity. Depending on research progress, we expanded our work to include additional agents for these two weeds, a seed-feeding caterpillar on tumbleweed and a flea beetle on yellow star thistle. When sufficient information was collected, we prepared and submitted a petition to USDA APHIS to request permission to introduce the new agents. When permission is granted, we will multiply and release the agents. We provided CALTRANS with information regarding the detection, identification, and use of the currently established agents and any new agents for yellow star thistle and tumbleweed. Right. I'm going to skip the achievements. But I may do a follow-up video or I may comment on them in the description, who knows. Mostly because when I went through this the first time, although they had some really interesting details, I don't think that's important for the main concern and reason and catalyst for this video. So there's a lot of good information in here. None of it has to do with the disapproval or the disapproval of that rumor, which I guess I'll call the Broad Mite and Hemp Russet Mite government rumor. That's not a very catchy name. Let's see here. Project design. The following is a brief description of the procedures. See appendices for more details on background and methods. For those of you who are interested in this sort of information, I'll make a follow-up video about the project design. But generally speaking, they used pretty standard evaluation techniques for regarding biocontrol agents. I'll briefly go over some parts of this. Essentially, here we have the weevil, which is Ceratapion barsicorna, and they tried to evaluate whether or not a fecund female would oviposit on non-target but related species of plants that they were trying to target for. And so here you can see two figures. Here you can see a representation of the adult female and some of the damage that the weevil does. Good luck trying to identify the weevil from this picture. Weevils, if you didn't know, are the most diverse group of beetles. And beetles are the most diverse group of insects. And insects are a huge representation of animals. So it can be difficult, to use the term lightly, to identify weevil species in general, especially since some of them are host specific and others are not, and you just really don't know. Some of them don't have that sort of characteristic nose either, so that can be very difficult. Sometimes they all just look like little small brown beetles and you can't really tell the difference without like expert identification or a look at their genitals. Usually, or a lot of times in cases where species are morphologically identical, the only morphological difference is their genitalia, because that's what keeps them from mating with other species. Although even then, that's not always true, but that's a story for another time. Here we have an example of one of the host specificity tests in a sleeve box. I won't go over it very much here. Here we have a host specificity evaluation of the tumbleweed agents. So I'm going to go into more detail on this one because this concerns Aceria Salsolae, which is the blister mite, the Eriophyid, that I think a lot of people are misconstruing with Hemp Russet Mite. So the paragraph reads, research focused on the mite Aceria Salsolae, which develops in leaf shoots and buds of tumbleweed. This mite was collected in Greece and evaluated in the USDA ARS quarantine laboratory in Albany, California. We had to develop methods to rear and evaluate the mite under containment so that it could not escape quarantine. We obtained seed of non-target test plants, many of which are native species. For the latter, we often had to develop methods to germinate and grow the plants. Mites were tested under no-choice conditions, in which an infested sprig of tumbleweed was put on a test plant. After 5 weeks, the test plant was examined under a microscope and we extracted any mites using a procedure that involved washing the plants and capturing the mites on a filter paper. Alright, so there's the blister mite. That's kind of what that's basically a small summary of what they did to evaluate the plant, or evaluate the plants specifically, and the mites' interaction with those plants. We have this figure here. It says field experiments were conducted at 3 sites in eastern Turkey during 3 years to determine if Ceratapion barcicorna, so this is not about the mites, attacks safflower under field conditions. The weevil was never reared from safflower despite high infestation of nearby yellow star thistle plants. So that's something that you want, right? This weevil will not go after plants even though they're right next to the crop, because it has a host, it has specific plants that it wants, and it's not those crop plants. There certainly is the plausibility, because the test is not exhaustive. It does not account for every single crop, every single plant, but they did narrow down the ones that they thought were most important, and it seems like the ones that they were testing for were not, especially safflower, which is an economically important crop, well, was not affected. So good on them. Here we have on the 8th page. Oh, and here we have this picture. So let me just keep that on here for now. Here's a summary of the impact for the yellow star thistle agent, which is the weevil. Impact of the weevil, Ceratapion barcicorna, was evaluated for infesting potted yellow star thistle plants and rearing them inside the quarantine greenhouse until the adult insects emerged. Plants were dried and weighed at the end of the experiment. Impact of the mite, Aceria Salsolae, was evaluated by infesting potted tumbleweed plants and rearing them inside a sleeve box in the quarantine mite room for 14 weeks. Plants were dried and weighed at the end of the experiment. And now we've gone to page number 9. Page number 9 is the page that I think is most responsible for people overreacting, if you could really call a picture responsible for somebody's assessment. But here we have it. Figure 5 and Figure 6. Figure 5 is an illustration and scanning electron micrograph of the Salsola Mite Aceria Salsolae. And this is from a paper, ostensibly. And you can see that there's a vermiform Eriophyid mite right there. And people are going to see that, and they're going to see some of the similar damage in Figure 6, which is the damage caused by Aceria Salsolae. And people, I think, have made this sort of presumption that this is Hemp Russet Mite. Unless you're an acarologist, if you're somebody who studies mites, or if you're an entomologist who has familiarity with that, or if you're some sort of a biocrop consultant, or anything like that, if you're a taxonomist, you would have a respect, perhaps a greater respect and a greater knowledge base of domain knowledge for understanding the subtle differences between different families, different species, different genera. Yeah, I don't know. I feel like, if you should be able to tell it's not Hemp Russet Mite by the name alone, even if you don't know those things. But, if somebody didn't read necessarily the report up to this point, they might not be aware of that. And hopefully this video can serve as a reference for people who are trying to understand with greater clarity what this report is all about. And in my opinion, this sort of demonstrates the incorrect nature of the rumor that Hemp Russet Mite is being spread, especially when people cite this report. Because while it is technically true that you can't prove a negative, this is not technically evidence that CALTRANS did not do something like that. This is not evidence that they did. So there you have it, right? Let me go to the description here for Figure 6. Damage caused by Aceria Salsolae, the mite. It looks like it says mite feeding kills meristems of Salsola tragus. So Salsolae, species name. Salsola is the genus of the Russian thistle, right? Salsola tragus. Comparison of uninfested cutting of Salsola tragus is on the left to a severely stunted. On the left in the right picture, in the picture on the right. So on the left we have one that is not infested, and on the right we have one that is severely stunted and infested. And these were collected in Greece. And anyone who's had to deal with Eriophyid mites like tomato russet mite, hemp russet mite, aloe mite, these are all similar mites that have sort of a similar effect. They've got phytotoxic, phytoreactive saliva. They've got compounds in their saliva that will gnarl the plant growth. And they create changes in the plant physiology, and it's very evident from this picture that this does look like that kind of damage. And if you were to look very closely, you could see the colony kind of erupt all over the plant material. But you can't see in this picture, obviously. Alright, so Figure 7. Figure 7 says, the caption says, test plants were infested with the Salsola Mite for 4 weeks. Plants were then examined under a microscope for damage and mite survival and reproduction. Any mites were extracted from the plants with a soapy solution and counted. Project results and discussion, foreign exploration and evaluation, BBCA conducted. They referenced BBCA earlier. It's a nonprofit organization that was affiliated here. The BBCA conducted exploration trips in Turkey, Kazakhstan, and Tunisia to discover new agents for tumbleweed. The weevil Larinus bilaformis was discovered attacking yellow star thistle, flowerheads in eastern Turkey, and Levant Gultekin. Oh, I see. This is a person's name. Levant Gultekin of Ataturk University conducted preliminary studies on its biology. Many new prospects for tumbleweed were discovered during the first trip into Kazakhstan in 2004. The leaf-feeding weevil, oh gosh, I don't know this name, Anthiperinus bimpressus, kind of a cool name, was discovered in Tunisia, and BBCA is beginning to conduct preliminary studies of its biology. So that has nothing to do with the Salsola Mite, but even when they had a couple of good agents, they still continued to try to find other ones because, in my opinion, it makes a lot of sense. Biological controls, sometimes they fail in certain temperature ranges, sometimes they fail in certain climates, sometimes they fail under certain exposures to pesticides and fungicides. Some things are more sensitive to certain things than others, and so having more tools in your toolbox is always a better situation to be in, right? It gives you more options to work with. This mostly goes over the weevil, and I want to sort of limit my coverage of the weevil specifically because it's not relevant to, like I said, the catalyst for this video. This is a table of species of insects found on tumbleweed in Italy, Kazakhstan, Tunisia, and Turkey. And I will go over this here because let me explain it this way. So here we have on the top we have Coleoptera, which is beetles, right? And we have Curculionidae, which are weevils, and we have a list of weevils here. We have coleopterans that are Chrysomylidae, which are leaf beetles, and there's only 2 species here. And we have Lepidoptera, which are butterflies and moths, and Pyralidae, which is a particular group of lepidopterans. So there were several insects found on tumbleweed in these 4 zones. These 4 zones are, I assume, places where, these are places where the tumbleweed was found, and so the idea is that this is how a lot of biocontrol is sort of found. If you ever wondered where Amblyceus swirskyii comes from, or any of the Amblyceus genus, they all come from different places and they can be found in different places, but they obviously are endemic or native to some zones. And so if you find them eating the pests that you're worried about, you can take those organisms and culture them and evaluate them and then use them as biocontrol agents like we have already. This is taxonomical stuff. It shows the sort of the taxonomy of different plant species here, and we're looking at the figure. The caption here says this is the mean number of Ceratapion barsicona eggs times 10, and the mean number of adult feeding holes per day. So this was an evaluation, it looks like, of basically the control factor of the weevil, but I'm going to skip that. Here we have another graph related to that. These are non-target species that were part of the choice oviposition experiment. So there were no choice experiments and there were choice experiments in sleeve boxes, right? And so these are all different kinds of, it looks like we have several species of knapweed. That would be the Centaureae genus. These were non-target organisms, and you can see that the last one, Centaureae solstitialis, obviously was the most popular one, but we did get some on Cyanus, right, and Melitensis, And we also got some on a totally different genus, Salsoria americana and Carthamus tinctorius. So to some degree, there seemed to be some egg laying on non-target species, right? But primarily, it did go and infest heavily the targeted species, which is what you want. There were safflower varieties, different varieties here that have sort of alphanumeric names, but compared to yellow star thistle, oviposition was heavily in the favor of the star thistle, right, for the weevil. Now we can go into the host specificity and evaluation of the tumbleweed agents. Obviously, the Salsola Mite was the most important, and it's the one that was primary in this article, but there were obviously other agents, right? Paragraph says, no live mites were found on any of the non-target test plants outside the genus Salsola after 4 weeks in laboratory experiments, and none of the non-target plants showed any sign of feeding damage. Nativity of plant species is indicated in Appendix 2 within Appendix 2 within Appendix 2. That's kind of funny. All positive control cuttings of Salsola tragus type A were infested with high numbers of mites, indicating that all the test plants were adequately challenged. Acerla Salsolae successfully reproduced on Salsola tragus type A, type B, type C, S. paulsenii, I assume, S. paulsenii, lax form, S. paulsenii spinosiform, and S. colina, all of which are considered to be noxious weeds, and all of which are in the Salsola section Cali, subsection Cali. That was a lot of taxonomic speak that even I didn't understand all of, mostly because I don't know what the different types are, but I understand that there are different varietal or sort of species complexes that kind of arise, and I assume that that's what they're referring to here. That's what happens when you skip parts of the report, I suppose. So sorry about that, but you can definitely take a look at the other sections yourself. And if you ever have any questions about understanding that, give me some sort of a message, comment. I'd be happy to go over it with you. Other experiments and field observations in Europe indicate that the mite also attacks S. cali. The mite could not reproduce on Salsola soda in the same genus, but more distantly related. Halogeton glomeratus, different genus but same tribe. Suaeda calceoliformis and S. mokinii, different tribe but same subfamily. Or Sarcobatus vermiculatus, another tribe but same subfamily. The reason why they're going over this and why this is relevant information, and I'm going over this with you, is to sort of illustrate that the Salsola Mite, or that the Salsola Mite has a very narrow host preference. It's not even going to certain species in the same genus. Going outside the genus is pretty unlikely that it will go after a plant that's not closely related to its target. And that's very important because that's what you want in a biocontrol agent. Last sentence, the small number of mites extracted from Salsola soda, Suaeda mokinii, and Gomphrena globosa all appeared to be dead. So it looks like that perhaps they were able to do some damage in the interim when they went onto those plants. Not very many did, and it seems like they died. And ostensibly, these experiments did not have examples of other organisms that could predate on the mites. So it seems like it's really toxic, perhaps, for them to feed outside of this genus even. So I don't think that, I mean, obviously the Salsola Mite is not Hemp Russet Mite, but not only that, I don't think that this plant or this organism can have cannabis plants as a host. So table 2 here, infestation of test plants 4 to 5 weeks after transfer of at least 20 mites. We've got a bunch of different names here, and I'm not going to try to pronounce all of them, but you can see that there's an evaluation of number of mites per infested test plant and a rate of positive control, which I might need more context to sort of explain. I don't really have that, but what you can see, and what I do understand from looking at this table, is that they tested for sugar beet, table beet, Swiss chard, rhubarb. I don't recognize any of the other scientific names off the top of my head, although it looks like we do have spinach, which is Spinacula oleracea. Yeah, so so they definitely looked at different plant species that could be affected, and it looks like they have some positive control variable that I'm not really aware of, or I don't understand very well, honestly. So there you go. Here, table 2. Oh, it's continued. So there's other plants here. We have, like we were saying before, there's a bunch of things in the subfamily of Salsoloideae. All of the Salsola genus plants, right? We can see that they didn't do very well on those plants. There's also Amaranthaceae. Okay. Huh. Yeah. So these tables are this table here and this table here. They're infestation. They're infestation tables, and what they're trying to articulate in this table is the amount of mites after a transfer of 20 mites. How many there were after 4 to 5 weeks. So, for example, the table beet, specifically the red ace hybrid down here, at the bottom of the first part of table 2, we have an example of 134 mites, I think. Yeah, I guess so. That must be what they mean. Even I'm getting confused at this table, honestly. So if there were more mites, one would surmise that they were able to exist on these plants. If I'm interpreting this correctly. I might not be. So, don't take my word for it on this table, specifically. But don't take my word for any of this. Do follow-up research. I would be curious to see if they were able to meet their goals. And this report obviously doesn't mention if they even decided to use this Eriophyid mite as control. So I don't really know personally. Here we go back to the weevil. I'm going to skip that. I'm going to go to the tumbleweed agent impact. In the quarantine experiment, mite populations established on all of the infested Salsola tragus plants, and signs of damage appeared within 4 weeks. The mite population increased exponentially during 25 weeks. Infested plants were 34% as large as uninfested plants. Based on combined length of all branches at 25 weeks after infestation, infested plants had 41% as much biomass as the uninfested plants. None of the plants had started flowering, so no impact on seed production was observed. The experimental light intensity, which is 140 micromoles per second photosynthetically active radiation was much lower than natural outdoor light, which is up to 1900 micromoles per second, and the temperature and humidity were more moderate than typical outdoor conditions. So, the experimental plants tended to be lankier and less stiff than in the field. So far, we have not been able to grow plants to maturity in the confines of the mite quarantine room, which hampers our ability to measure impact on seed production, which of course is an important aspect of this. If you don't have enough seed, if you're able to impact seed production, then you're going to impact the ability for the plant to reproduce and obviously spread, right? In Europe, mites are found on dramatically stunted plants. In a field study in Afyon, Turkey, infested plants were less than half the size of uninfested plants. The maximum size of naturally infested plants was half that of uninfested plants at the end of August. We have not tested the mite's ability to impact Astragus type B, S. colina, or S. paulsenii for impact, but we'll do it as soon as we have improved our experimental conditions. In the absence of other data, we would like to expect the mite to cause similar or less damage to these and other Salsola species, and that does make sense. Here's a figure. Caption reads, population growth of mite Acerla Salsolae on Salsola tragus plants during impact study, which is this is number per 4 centimeter branch tip, plus or minus the SE. So we have a pretty nominal growth curve, to be honest. Here's a comparison of growth of uninfested Salsola tragus to infested plants on the right. Weeks after infestation of the mite Aceria Salsolae, grown under artificial lights inside quarantine laboratories. So visually, you can definitely tell the difference. On the left, we have uninfested plants, and on the right, we have infested plants. And it's right there. They're much smaller. I don't know what else to say about that. Conclusions. Cool. We're finally on page 20. I'll go over the tumbleweed mite part first, and probably only, in fact. The tumbleweed mite, Aceria Salsolae, is able to feed and reproduce only on a few closely related species in the Salsola section, Cali subsection Cali, including Salsola tragus type A, Salsola tragus type B, Salsola Cali, Salsola paulsenii, and Salsola colina. It was not able to reproduce on or damage any of the 36 non-target species tested, including Salsola soda, which is in a different section of the genus from Salsola tragus. The mite stunts the growth of tumbleweed and should reduce plant size by 34 to 50% and probably reduces seed production. The reason why they say that is, in my opinion, obvious, but to explain why I feel that way, obviously, a lot of times significant plant damage, especially, and localized plant damage to flowers and fruits and things like that, definitely hinder and impair reproductive ability of a plant. So you would think that with like a 50% reduction in plant size, the reproductive ability of that plant will be affected in some capacity. We believe that our results are sufficient to demonstrate that the mite is safe and likely to impact tumbleweed. We submitted a technical advisory group petition to the USDA APHIS in December of 2004. TAG gave it a positive review, and we submitted a formal request to USDA APHIS for a permit to release Aceria Salsolae. This is a major step towards obtaining a regulatory permit to release a new agent for tumbleweed, Russian thistle. Such an agent is expected to provide self-sustaining long-term reduction in plant size and seed production of tumbleweeds. This should help reduce the amount of herbicide applied for its control and reduce the occurrence of tumbleweed-associated automobile accidents, which I'm guessing is a non-zero number. So for those of you who think that's silly, I don't think the people in accidents think that's silly. So this research has made significant advances to the discovery and development of new biological control agents for yellow star thistle and tumbleweed. Petitions have been submitted for 2 agents. Research has also been conducted on 2 follow-up agents. That would be the Psiloides calcomera for yellow star thistle and the Gymnansila cannella for tumbleweed. Discovery of beetles attacking tumbleweed in Kazakhstan and Tunisia greatly increase our chances of finding an agent, I would say an agent, that will successfully control tumbleweed. They talk about future work, which has to do with these new organisms. They say down here that release of Aceria Salsolae, and one of the things they want to do is release Aceria Salsolae, obviously, and evaluate its impact on tumbleweeds, which cannot be done until permit is issued, possibly in 2007. I don't know. And then we have here references. There's a ton of references. There always are. I'd like to point out here, since it caught my eye when I was reading, but we have here this article titled Preliminary Studies on the Biology and Host Specificity of Aceria Salsolae. It's Acari Eriophyid, so it's a mite. It's an Eriophyid mite. It's an Eriophyid mite. That's what it is. So it is an Eriophyid. It is not russet mite. It's not hemp russet mite. It's not tomato russet mite. It's not aloe russet mite. It's a different russet mite. And maybe it affects cannabis plants. Maybe. Someone would have to have an acarologist kind of identify it for you, who's, first of all, specialized in Eriophyids and has a key and can actually do the identification, because Eriophyids can be morphologically really difficult to parse, to be quite honest. So yeah, so probably not a worry for cannabis going into the future. Yeah, I mean, based on this report, there's really very little reason somebody would think that the Salsola Mite would be able to affect cannabis plants. Like I said earlier here, the mites, no live mites were found on any of the non-target test plants outside the genus Salsola after 4 weeks. So that includes cannabis plants. And obviously nowhere in this report did they refer to Polyphagotarsonemus latus. They did not refer to Aculops cannibicola, which would be the Broad Mite and Hemp Russet Mite, respectively. Yeah, that's all I wanted to say. It's been like an hour. If you have any questions, any comments, any follow-up, any reports that you want to send my way regarding this or similar topics, I would be very interested in reading them. I would be very interested in looking at that sort of thing. I think the first thing I'm going to do after this is look myself for follow-up and report on it as necessary. Thanks for watching if you have. If you like this kind of information and this sort of in-depth reporting, feel free to like this video, give me a comment, subscribe for more. I will probably be doing more commentary like this in the future. I have a dicamba video that I made recently that is that talked about the big dicamba incident in soybean crops, and I kind of took that as a way to as a avenue to talk about volatility in pesticides in general, because I feel that's very underrepresented in pesticide use in general. So that's it. Have a good one.